Welcome to the Physiology Odyssey. So today's lecture is about blood and plasma proteins. So here, what is the importance of blood we will know in this lecture, and what are the functions of plasma proteins. And here in MBBS first year, there will be lot of questions from this blood and plasma proteins, and there will be viva, and this topic is important for your practicals also. So let us start here. So after this lecture, the student will be able to know or must know areas. So that is, what is the blood? Define blood and give the composition and function of blood. Then name the layers of blood after centrifugation, name the cells present in Buffy coat, and give uses of Buffy coat preparation. So what is the Buffy coat? We will know in this lecture. And give the normal value of value of blood volume in the adults and children, and describe the methods of blood volume measurement, and give the composition and function of plasma, name the plasma proteins, give their normal values, and list the functions of plasma proteins. So here there is a short note on this topic: name the plasma protein, give their normal values, and list the functions of plasma protein, give the uses of fresh frozen plasma. So next slide. Blood. Blood is defined as liquid connective tissue that fills the heart and blood vessels. So it is a liquid connective tissue. What is the normal volume? 5 to 6 liters in adults. So composition of blood, it has two components, that is cells and fluids. And cellular component consists of different form elements, and fluid component is the plasma. So form elements are red blood cells, erythrocytes, white blood cells, leukocytes, and platelets, that is the thrombocytes. So these are the form elements. So another name for red blood cell is erythrocytes. So erythro, it is a Greek word meaning red, and leuko, this is a Greek word, and what is the meaning? Meaning is white. Leuko means white. And another name for platelets, this is thrombocytes. So this is the appearance of centrifuge blood. Note, about 1% of blood from the Buffy coat. So this Buffy coat, after we centrifugate the tube at a very rapid speed, so there will be settlement of these red blood cells in the bottom. You can see this red layer, this is the red cells. So 43 to 47% is red cells. Then plasma, you can see the upper layer, it is plasma. So here, what is the percentage of plasma? 52 to 57%. And between this plasma and red blood cells, there is this yellow colored layer. This is called Buffy layer. And what is the constituent of Buffy layer or Buffy coat? WBCs and platelets. So here, there is a multiple choice question here. What is the constituent of Buffy coat? So WBCs and platelets are the constituent of Buffy coat. So what is the importance of this Buffy coat? What is the clinical importance? So in infectious conditions, this layer Buffy coat will be thick. Then plasma. So plasma consists of about 55% of the total blood volume, which is made up of water and solid particles. And then 45% will be form element, that is RBCs, WBCs, and platelets. When blood is collected in an anticoagulated tube and allowed to settle by centrifugation, three layers are distinctly visible. So the upper plasma layer is separated from the lower red cell mass by a thin Buffy coat. So here, this thin middle layer that separates the upper plasma and lower red cell mass after centrifugation of the blood is the Buffy coat. It contains leukocytes, that is WBCs, white blood cells, and platelets. So here, clinical box, this is what is the importance. So here, blood coat preparation. The blood film may be prepared from Buffy coat for the detection of abnormal cells in the body, in the blood. Also Buffy coat preparation is very useful for the detection of bacteria, fungi, or parasites within neutrophils, monocytes, or circulating macrophages. So in certain infectious diseases, there will be appearance of this abnormal cells, that is bacteria, fungi, or parasite inside the neutrophils, monocytes and circulatory macrophages. So this is the importance of functions of blood. Now functions of blood: respiratory function. Blood transport oxygen from lung to tissues and carbon dioxide from tissues to the lung. So this is very important because our all organs and tissue requires constant supply of nutrients from GIT. So here this blood, it contains nutrition like, for example, glucose, amino acids, various hormones and then vitamins. And this blood, it will transport the oxygen from lungs to tissue and carbon dioxide from tissues to the lung. Then transport medium. Blood acts as a transport medium for various hormones, chemical substances, nutrients, vitamins, etc. Hormones secreted from various endocrine tissues and nutrients absorbed from GIT tract circulate in blood and distribute to all tissues of the body. So thus blood regulates growth and metabolism of tissues. See, the growth and metabolism of tissue, it depends upon the constant supply of blood. So blood, it is having the important function of transport. So it will transport the nutrient substances like glucose, amino acids, fats, vitamins to the different tissues of the body. And apart from this, it will collect the waste material like carbon dioxide and lactic acid from the tissues, and it will then this blood, it will goes to the lung, and from lung this carbon dioxide will be excreted from the body. So temperature regulation, this is another important function of blood. Blood conducts the heat from the interior of the body to the surface through blood vessels. Temperature is continuously produced in the body. Blood absorbs this heat and transfer to the body surface. So for the survival of our body, the temperature regulation is very, very important, and this blood, it is helping in the regulation of temperature. Excretory function, blood helps in the excretion of waste material by transporting them from different parts of the body to the kidney. So this is very important here. Water homeostasis. What is homeostasis? Keeping the balance in the body. So all here homeostasis means keeping the things in the constant level. So here homeostasis of the water, it is maintained by blood. So as the blood contains 55% of the here in blood 55% plasma is there, and out of this 92% of plasma contains water. Loss of water from body as occurs in diarrhea, vomiting, excessive sweating, etc. result in decreased blood volume of the blood volume. So this is called hypovolemia. So persistent hypovolemia result in cellular dehydration. See, for the survival of our all cells, the constant supply of water is also very important. Otherwise there will be dehydration of the cells and ultimately cell will die. So blood, it is maintaining the level of water in the body. So this is one of the another important function of blood. In response to hypovolemia, mechanisms are activated to increase the water content of blood that aims at maintaining cellular hydration. So here, whenever there is excessive sweating or excessive vomiting or diarrhea, there will be loss of water from the body. This will lead to hypovolemia. And this hypovolemia or decrease in the volume of blood, decrease in the volume of water may severely affect our body. So here, whenever there is loss of water or loss of blood from the body, there will be hypovolemia. So here, different type of negative feedback mechanisms, they will be activated to restore the blood and water in the body. For example, more ADH will be secreted and kidney will absorb more water. So here the blood, it is having important role in the homeostasis of water. Then acid base balance. Blood contains plasma proteins and hemoglobins. They play important role in maintaining acid base balance of the body. Also bicarbonate in the blood buffers the acid. So here importance of maintaining acid base balance is there. And immunity. Blood forms a critical component of body immunity. It contains cells that play primary role in cellular immunity, also contains antibodies that play major role in humoral immunity. So here then blood is a storage site of electrolytes, nutrients, chemicals and hormones. So blood, it also act as a storage function. It has storage function. So here, blood color, body provides natural color to the body. So blood it provides natural color to the body. In anemia, in hypovolemia, body becomes pale. So blood, it gives a particular color to our body. Then nutritive function, blood delivers all nutrients such as glucose, lipids, proteins, vitamins, minerals, etc. to all tissues of the body for their utilization and storage. Oncotic pressure. Oncotic pressure. Oncotic pressure, the albumin present in the blood exerts osmotic pressure known as oncotic pressure that controls capillary filtration and prevent edema formation. So these are the different important functions of the blood. So here we are discussing about then total blood volume. Total blood volume can be roughly calculated as 70 to 80 ml per kg of the body weight. Here there is a MCQ. So total body volume can be roughly calculated at as 70 to 80 ml per kg of body weight. This bold colored is highlighting the MCQs. So here 70 to 80 ml per kg of the body weight is the total blood volume. In adults males, the total volume of blood is about 5 to 6 liters, and in females it is less. It is 4.5 to 5.5 liters. In children, though the absolute volume of blood present is significantly less than the adults, so it is also expressed per square meter of body surface area, which is normally 2.8 liter per square meter of the body. So this is also a MCQ here. So 2.8 liter per square meter of body is the per square meter of the body surface area. This is blood volume. It is expressed like this. Determination of blood volume is determined by plasma volume and determining the cell volume. So the normal plasma volume to cell volume is 55 by 45. So 55% of total blood volume is plasma and 45% of the total cell volume is there. So here we can see. Then measurement of cell volume. Cell volume is measured by measuring the volume of red cells. So red cell volume is the volume occupied by the all circulating red cells. So it is usually calculated by subtracting plasma volume from the total blood volume. It is also directly calculated by injecting tagged red cells. The radioactive isotope of chromium is usually used. Determination of plasma volume. So here these are the MCQs. Plasma volume is usually measured by using dyes. What is the name of that dye? That is, the dye used is Evans blue T 1824. This is multiple choice question here. So Evans blue is used here. It is also measured by injection of serum albumin labeled with radioactive iodine. So this is also a method. An average plasma volume is about 3.5 liter. 5% of body weight of a 70 kg man. So here these are all MCQ. Most important is for the determination of plasma volume, one dye is used. And what is the name of that dye? Evans blue T 1824. The total blood volume is calculated by multiplying plasma volume with 100 by 100 minus hematocrit. This is the formula for the calculation of total blood volume. So for example, if hematocrit is 40 and plasma volume is 3.5 liter, then blood volume will be 5.8 liter. So we are multiplying 3.5 into this formula, 100 by 100 minus 40. So we have to distract here 40 from the 100. Effective blood volume is equal to total blood volume minus the volume that is sequestrated. This means the volume which is present actually in circulation that helps in perfusion of tissue. So this is the effective blood volume, means which is helping in the perfusion of tissue or supply of blood to the tissue. Specific gravity and viscosity of blood. Specific gravity approximately 1.050. This is MCQ. This depends on the number of cells and composition of plasma. Specific gravity is measured by extent of light bending in a refractometer or is a part of test strip dipped into blood. Usually loss of water from blood as occurs in dehydration increases specific gravity. So in conditions like severe diarrhea or vomiting, there will be increase in the specific gravity of blood. Viscosity of the blood is measured of resistance to flow. See, there is a MCQ here, multiple choice question is there. If the viscosity of blood increases, so there will be, if the temperature of blood increases, there will be decrease of the viscosity. If there is increase in the temperature of body, there will be decrease in the viscosity. There is opposite relationship is there, reciprocal relationship is there. Blood viscosity is 4 to 5 times that of water. So blood viscosity is more than of water. It is mainly due to the number of cells and macromolecules like protein present in the blood. Then what is plasma? Plasma is a fluid component of the blood which constitute about 55% of total blood volume. This is MCQ. For 55% of the total blood volume is plasma. The remaining 45% is constituted by cells or form elements like RBCs, WBCs and platelets. So here if the whole plasma is to be used for investigations, blood is collected with anticoagulated blood. It can be stored for years for future investigations. So here what is the composition of plasma? 92% water, solid or solids 8%. Plasma proteins makes 90% of the solids present in the plasma. Inorganic means anions. Anions means negatively charged anions. They are called anions. So they are, for example, chloride, bicarbonate, phosphates, sulfates, etc. Then cations. Cations means positively charged ions. For example, sodium, potassium, calcium, magnesium, etc. Organic, that is colloids. Plasma proteins colloids, and crystalloids, that is glucose, lipid, urea, uric acid, etc. Once again there is a MCQ. See, which are the crystalloids present in the blood? Glucose, lipids, urea, uric acid. But plasma protein is not a crystalloid, it is colloid. For example, albumin is a plasma protein. So albumin is a colloid, it is not crystalloid. So here we can see the different MCQs from this slide. Then constituent of plasma, note 92% of plasma is water and 2% is solute. So here 92% of water and 7% is proteins, and albumin it is 54%, globulin it is 38%, Fibrinogen it is 7% and other 1%. So major constituent of plasma protein is albumin, followed by globulin, then Fibrinogen. So oncotic pressure, that is the pressure because of plasma proteins, it is called oncotic pressure, and mainly the albumin is responsible for the oncotic pressure due to plasma protein. So this is a MCQ or multiple choice question. Albumin is responsible for the oncotic pressure exerted by plasma proteins. Now what is fresh frozen plasma? There may be a brief answer question. So here in some condition plasma need to be stored for years for future analysis, for which plasma is stored frozen below minus 20 degrees Celsius. You have to remember this. This is called fresh frozen plasma, which can be stored up to 7 years in a blood bank. So here what is the importance of this? To preserve clotting factors, plasma should be frozen within 6 hours after collection. This fresh frozen plasma is used for plasmapheresis. So this is a process, therapeutic plasma exchange. In certain diseases, this fresh frozen plasma is stored, used. See, what is fresh frozen plasma? It is stored for years in a frozen style, means in minus 20 degrees Celsius. This is called fresh frozen plasma, which can be stored up to 7 years in a blood bank, and it is used for therapeutic conditions or therapeutic plasma exchange. Some diseases require the exchange of plasma. Serum. What is serum? During blood coagulation, a soluble plasma protein called fibrinogen is removed from the plasma, and it is utilized in the formation of fibrin, that is the clotting. So plasma without this clotting factor is called serum. Serum, it is nothing but the plasma without clotting factors. So many biochemical tests are performed by separating serum from blood by allowing the blood to clot, coagulated blood. So serotonin content of serum is high as platelets release serotonin during clotting. It is not only used for biochemical investigation but also as a supplement to cell culture media. It ensures a better environment for growth of cells in vitro as it contains all nutrients, sugar, proteins, hormones and other factors. Fetal calf serum is used often for the growth of human cells. Separating serum plasma is separated from centrifugating anticoagulated blood. The serum is separated from blood by allowing the blood to coagulate. So serum is equal to blood minus fibrinogen and other clotting factors. So blood without fibrinogen and clotting factor is called serum. Then plasma protein. This is a very important part of this lecture, as there is very frequently asked question on plasma proteins. So you have to write down the different types of plasma proteins, their composition, their functions. So plasma proteins are the major solute constituent of plasma. The normal plasma protein concentration is 6 to 8 gram per 100 ml of plasma. So here there is a MCQ. What is the normal concentration of plasma protein? It is 6 to 8 gram per 100 ml of plasma. Plasma proteins are of three types: albumin 4.4 to 5.5 gram per deciliter, globulin 1.5 to 3 gram per deciliter, and fibrinogen 0.3 gram per deciliter. These are all MCQs. You have to remember the concentration of albumin, globulin and fibrinogen in the plasma proteins. So this is what is the concentration of albumin? It is 4 to 5.5 gram percent, or globulin 1.5 to 3 gram percent, and fibrinogen 0.3 gram percent. So fibrinogen is less here. Plasma proteins are formed mainly in the liver. So here, which organ is responsible for the production of plasma protein? It is liver. Then types. Albumin first. This is the major constituent of plasma protein. Albumin is formed in the liver. So it has a half-life of about 20 days. Being smallest in diameter among the plasma protein, in kidney diseases with glomerular injury, it appears early in urine. So appearance of albumin in the urine, it is called albuminuria. So whenever there is damage to the kidney, this albumin being small in diameter, it will pass through the kidney and it will appear in the urine, and this is called albuminuria. So the appearance of albumin in the urine is a symptom of a kidney disease. So whenever there is damage to the kidney, there will be appearance of albumin in the urine, and this is called albuminuria. Appearance of albumin in urea is called albuminuria. In urine is called appearance of albumin in the urine, it is called albuminuria. Globulin. This is formed in the liver, cells of reticuloendothelial system and plasma cells, divided into three categories: alpha, that is alpha 1 and alpha 2, beta 1 and beta 2, and gamma. So globulin is a globulin. This is the another important plasma protein after albumin. So molecular weight is 90,000 to 1,56,000. They include different transport proteins like transferrin, ceruloplasmin and hemoxin. They form different lipoproteins in combination with lipids in plasma. So So these are the functions of this globulin. So it will help in the transportation of iron. So it is called transferrin, and ceruloplasmin and hemopexin. Then globulin. What is C? Another function of globulin continues here. It will help in the formation of antibodies. They are called gamma globulin that are formed by plasma cells. So the normal albumin globulin ratio is 1.7 by 1 or 1.2 to 2.1 by 1. So you can see here that this 1 is constant. So this is MCQ here. What is the normal ratio of this albumin to globulin? In many diseases, especially in immunological and inflammatory diseases, AGR is altered. Detection of AGR help in diagnosis and assessing the prognosis of some diseases. So albumin globulin ratio is important. Then fibrinogen. It is produced in the liver. It is also a plasma protein. Fibrinogen is a plasma protein and it is produced in liver. It plays an important role in blood coagulation. It also contributes to the viscosity of plasma and determination of ESR. So these are the importance of this different plasma protein. Now the most important part is coming, that is the function of plasma. What are the functions? So osmotic pressure. The plasma proteins are osmotically active molecules, and the osmotic pressure of plasma due to plasma protein is called oncotic pressure. So the normal oncotic pressure is 25 mm of mercury. This pressure helps in maintaining volume of vascular compartment. So oncotic pressure retains fluid in the vascular compartment and therefore prevent loss of fluid from capillaries into the interstitial tissue space. Functionally, it opposes the action of hydrostatic pressure. The oncotic pressure is due to the presence of albumin in the plasma. See here, this is a very basic concept. If this plasma protein, especially the albumin, it is responsible for the oncotic pressure and it will retain the fluid inside the blood vessels. If there is decrease in the production of albumin, the fluid will pass from blood vessel to the interstitial space, and because of the leakage of fluid there will be swelling of the body, and this will produce the edema. So in certain diseases of liver there will be for example in liver failure, the liver will be not able to produce the albumin, and because of this the oncotic pressure of this blood vessels will decrease and there will be leakage of fluid from the blood vessel into the different tissues of the body, leading to the swelling of the body. So swelling of the legs, swelling of the body, this is a very common symptom of liver diseases. This is because of the decreased production of plasma protein, especially albumin is not produced by the liver, leading to the edema or swelling of the body. This is the clinical importance of the plasma proteins. So important note here is anemia and edema are features of hypoproteinemia. In conditions of hypoproteinemia, decreased oncotic pressure result in edema. Hypoproteinemia means decreased concentration of decreased quantity of this plasma proteins in the blood. So it is called hypo. Hypo means decrease. Proteinemia. So protein content of blood is decrease in hypoproteinemia, leading to decreased oncotic pressure resulting in swelling or edema. As protein is needed for globulin part of hemoglobin synthesis, anemia is common in hypoproteinemia. Hence, these conditions are named as anemia and hypoproteinemia. So, whenever there is hypoproteinemia, there will be anemia. So, here decreased production of hemoglobin will lead to the decreased quantity of RBCs also, and there will be anemia. So, always hypoproteinemia will be accompanied by anemia in the patient. So, this is the clinical importance of these plasma proteins. Then, what is the role of plasma protein in viscosity of blood? Viscosity of blood is the resistance of any type of resistance in the flow of blood. It is called viscosity of blood. Whenever there is increase in the viscosity of blood, there will be increased chance of increase in blood pressure also. So here plasma protein contribute to about 50% of the viscosity of blood. Red cells account for the rest of the viscosity. The viscosity depends on the molecular shape of the plasma protein. This is why fibrinogen molecules that are elongated and fibrillar in shape contribute more to blood viscosity than albumin molecules that are ellipsoid in nature. So, shape of the different type of plasma proteins are responsible for the viscosity. Mainly elongated and fibrillar shape is there of fibrinogen, and this fibrinogen it is responsible for the viscosity of blood. Immunity antibodies are plasma proteins, gamma globulins. Antibodies mediate humoral immunity that protect the body from infections, especially extracellular pathogens and from the effect of toxic substances. So, this antibodies that are the plasma proteins is actually gamma globulins. They will help in the strengthening the immunity of our body. Blood coagulation, blood clotting depends on concentration of fibrinogen that forms fibrin thread, and final step in blood coagulation also other clotting factors. Prothrombin are plasma proteins. So, here we can say these are the different functions of plasma proteins. So, whenever there is short note on this, you have to write down all these functions. What is the role of plasma protein in viscosity of blood, immunity and blood coagulation. Then carrier protein. Plasma proteins serve as a carrier molecule for transport of various substances like hormones, drugs and metals. A carrier for water insoluble lipophilic substances, albumin acts as a carrier of steroid hormones, fatty acid and thyroid hormones. Specific barrier proteins, they are meant for the transport of lipophilic hormones to target cells. Examples are sex hormones binding globulin that bind to estradiol and testosterone thyroxine binding globulin disease. Buffering. The plasma proteins form an important buffering system of the body called protein buffer. This helps in acid-base balance of the body. This is another important function of this plasma protein. So, they will help in the buffering system. So, they will help in the maintaining of acid-base balance in the body. Then, lipid protein store. Plasma protein serves as mobile protein reserve as a source of amino acids for tissues for the body, which can be utilized for tissue growth, especially in situation of protein depletion. Synthetic function, plasma protein provide substrate for the synthesis of protein hormones like erythropoietin. This is very important. This erythropoietin, it is helpful and essential for the maturation of RBCs. So, production of blood or erythropoiesis requires the presence of this erythropoietin. This is a protein hormone, and plasma protein are important for the formation of erythropoietin. They will provide substrate for the synthesis of protein molecules like erythropoietin, etc. Various enzymes for synthesis of hemoglobin. Hemoglobin is very important. So, this plasma protein, they will help for the formation of hemoglobin also. Then, determination of ESR. What is ESR? Mostly depend on the concentration of fibrinogen in plasma. So, fibrinogen, it is also a plasma protein. So, fibrinogen facilitates Rolex formation, which in turn increases the rate of sedimentation of red cells. So, whenever this RBCs when settled, they have a tendency to stick to each other and form a Rolex or pile of this RBCs. This is called Rolex formation. Like when the coins stick to each other and form a pile, so it is just like that the RBC stick to each other. So, here this fibrinogen help in this condition in which fibrinogen concentration is more like acute inflammation, ESR become more. So, how this is a formation of Rolex formation, it is measured by this ESR, erythrocyte sedimentation rate, it is called. Separation of plasma protein. Plasma protein can be separated by methods like salt separation, paper electrophoresis, Cohn's fractionation and ultracentrifugation immunochemical analysis. What is plasmapheresis? It is a process by which plasma can be removed from blood without removing red cells. So, plasma is separated from blood. It is usually done to separate excess or abnormal plasma proteins such as antibodies from body. So, in various autoantibody diseases, this plasmapheresis is done. So, it is a process by which plasma can be removed from blood without removing red cells. So, So plasma is separated from blood and it is called plasmapheresis. So usually it is done to separate excess or abnormal plasma proteins. So here this is MCQ. What is the purpose of plasmapheresis? It is usually done to separate excess or abnormal plasma proteins such as antibodies from the blood. So this was from this video. In next video we will provide you with the solved MCQs. We will discuss the different MCQs on blood. So stay connected, subscribe our channel, share it. Thank you.